This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 19, recorded on October 21st, 2011. Hello, everyone. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent. How are you this afternoon? I'm coming to you from beautiful Boston.
That's right. What are you doing in Boston, Michael?
I'm at the Infectious Disease Society of America's annual meeting. That's this year in Boston. Next year it will be in Elio's hometown of San Diego. But this year it's in his old hometown of Boston, and we're presenting some of our initial results from our copper trial that we hope to submit in the next few weeks to a journal. And then once it publishes, we'll discuss it on TWiM. You can take me to task.
Excellent. We'll have to get some toughies on to ask you good questions, right? Also joining us today from Small Things Considered, Elio Schaechter.
Hi there. Always nice to hear your voice.
How you doing, Elio?
Great. Doing fine.
Everything's good in San Diego?
Yes, sir. No, nothing to complain about.
All right. Rejoining us today from Yale University, Joe Handelsman. Welcome back, Joe.
Thanks, Vincent. How are your microbes today?
How's your microbiome?
How's your microbiome?
It's
pretty regular.
I don't use artificial sweeteners.
Good.
But I do like red wine.
Oh, your microbes do too.
Apparently. What a great story. Can't wait to talk about that. Well, welcome back. Good to have you again. And today we have two really cool stories. The first paper is the sequence of a Yersinia pestis genome from victims of the Black Death, just published in Nature. And the authors, well, there are a lot of authors. The first author is Boss, and the last author is Kraus. And I think, Michael, you're going to tell us about this. Am I right?
Yeah, I get the lead on this, I guess, because I'm the one who has the honor of teaching dental students. And this was a remarkable paper in that it was remarkable for a large number of reasons. But first and foremost is they recovered PCR-grade nucleic acid from dental materials recovered from bodies that were buried in medieval catastrophe burial from the first outbreak of plague in 1348. So this is DNA from approximately 660 years ago. And what they did is they isolated the DNA from 46 teeth and 53 bones. And principally what they did is they took the roots and the crowns from the teeth and they pulverized them, and they recovered the nucleic acid from the roots and the crown of the tooth, and they were able to get sufficient DNA from which they were then able to use a baiting technology in which they used the current one of the current human pathogenic strains of Yersinia pestis, the agent suspected for causing the Black Death from 1348, and they used that as the bait. So they took this nucleic acid that they extracted from these teeth and bones, and they poured it, if you will, onto this array, and they were able to get the extracted DNA to stick, and then through the magic of PCR and array-based technology, they then amplified it up and determined the nucleotide sequence. And
Michael, can I ask you why they use teeth and bones?
They didn't make it clear as to why they used teeth and bones, but reading between the lines and then the supplementary papers, I think it's because they had access to the skeleton. And from 660 years ago, I don't think there's anything left of the body but teeth and bones.
But there's another reason, I think. These are sheltered sites in the body. And so bacteria aren't going to get into the pulp cavity of the tooth as easily as they would get into someplace else, even bones. So teeth have been used actually by quite a few people for this purpose. And in addition, DNA sticks to bone material, hydroxyapatite or whatever it is. And so this is a favored material because for the reason you just mentioned, that it's the only thing that's left of the body, bones and teeth, but also teeth are just a beautiful little capsule that encapsulates time.
So it's because, Elio, there was at one time blood in the teeth?
Right, exactly. So this only works for blood, for hematogenetically spreading bacteria or whatever, because anything that's not spread to the blood wouldn't be detected this way. But the plague is fine.
Yeah, the plague, again, that was where I was going with it. The second reason this paper was remarkable is they asked a very simple and straightforward question: is how similar was the DNA taken from these archival materials to the known virulent strains, ones that cause disease in humans. But before we get to the nitty-gritty of how they did this piece of science, we have to consider why they did it. There's been much debate about the utility of looking at genomic DNA and trying to answer questions as to beyond the so what. So you sequence a bunch of DNA, what does it really mean? And I think they're trying to develop the inferences of the importance of things, and that's what leads then to the significance of their data. But let's think a little bit about the pathogenesis of Yersinia pestis and how it does indeed cause disease in people. First and foremost is this bacterium is thought to have been responsible for killing 1/3 to 1/2 of the population of England at the time that it was running through Europe. And during this first wave of plague, it was thought to have taken out about 30 million people. And in England, it was thought to take out between 3 and 7 million people during the time. And so the time we're talking about is 100 years before Christopher Columbus, that we discussed last week, discovered the New World in 1492. And looking at what was going on and how this microbe actually spread, and it was thought to have come from China via the silk trade routes or via ships. And when it's moving over land, it's estimated, because this is again an accidental pathogen of man, it's principally a flea that bites a rat and takes a blood meal from the rat, and man is the accidental host of this flea. Typically what happens is the Yersinia literally stop up the works of the flea so it can no longer digest the blood, because the Yersinia literally becomes chock-a-block full in the gut of the flea. And so when the flea bites you, it regurgitates the contents of its gut into the new host. And man is the accidental host being bit by the flea, and that's of course how you develop the bubonic form of this. And the bubonic form of plague has about a 2 to 6 day incubation period, and you will present with a onset of a sudden fever and then extreme prostration. And if you're not medically inclined, extreme prostration means that you're too tired to text. If you look at the new generation of our students and our children who are constantly texting, extreme prostration means that you're too tired to text.
That's
and
graphic.
So
that was true even in the 14th century.
It's a good reference. And in addition, you end up with swollen, inflamed lymph nodes that develop the classic buboes. And hence the term boo-boo, which was an inflamed, swollen, necrotic lymph node. And mortality from the bubonic form of plague is about 25 to 50% mortality. And then as this microbe spreads through the blood, and as Elio so eloquently pointed out, it's principally distributed through the bloodstream, which is why they were able to recover it from bones and teeth. And so it's really one of these diseases for which we can do archiving and ask evolutionary questions about the evolution of pathogens and which genes are important that really cause this level of mortality. And once the plague bacillus gets into our lungs and we begin to go into the pneumonic stage and the acquisition, you principally inhale it in the pneumonic form, there's a 2 to 3 day incubation period, and then it effectively has 100% mortality. And so I think the author's reason for pursuing this sequence comparison is they thought the difference between the Black Death microbe and the current plague strains that are extant in the wild and in culture collections, they were then going to be able to compare and contrast and ask who's missing or what differences are present between the microbes of old and the microbes of present. And I think it was really interesting that they used, if you will, the strain that was responsible for the death of the individual to go on their, if you pardon the pun, their fishing expedition, because they were literally fishing the nucleic acid out from the extraction mix onto the modern comparator strain. And they then asked a very simple question, is how similar was the DNA from then to the DNA now?
Michael, can we ask you a question?
Please.
So here's a naive question. You use today's Y. pestis DNA to fish out DNA from the teeth and the bones. Well, what if there were some other pathogen present that exacerbated the Black Death? We wouldn't see it, right?
Okay, and so you're asking the question, if it wasn't Yersinia pestis that killed them, and there was some other microbe that was in the teeth and bones, would their technology be able to detect it? And since they were specifically fishing for Yersinia genomes, I don't think if there was some other microbe present, they could be able to detect it, because they were specifically hunting for the 4.6 megabase chromosome of Yersinia, as well as its two associated plasmids. So I think that's a limitation of the study. If there was some other secondary microbe that was facilitating the disease process, a co-infection, if you will, I think this method would miss it.
Okay, got it.
Let me also add, if I may, a little of the chronological perspective. It may sound like 400 or 600 years ago is not a lot of time, but it's been found that Yersinia seems to be a fast mutator. That is, it seems to have arisen from a soil bacterium called Yersinia pseudotuberculosis, which also infects people, but doesn't cause the Black Death. And this happened, according to Mark Achtman, in a paper he wrote about 13 years ago. It happened in prehistoric times. So these are not evolutions that take place over the hundreds of thousands or millions of years, but they happen within the thousands of year framework. So it makes sense to ask this question, because this is an organism that may have changed, that may be changing in front of our eyes, as it were.
Okay, Michael, what did they find?
Okay, what they found in their story is that the DNA sequenced from the 5 teeth via their molecular capture methodology, and they authenticated it first and foremost to, there's a method by looking at C to T damage that's characteristic of authentic endogenous ancient DNA. So the molecular biologists have worked out a method to determine how many hits piece of nucleic acid needs to take in order to determine that it's ancient. And what they found is they found very little differences between the ancient microbe and the current strain. They effectively came up and they justify it by going through their figures, showing that they have sufficient coverage and all of those other things. And what they found is that the ancient organism looks old. That's the first thing. And when they compared it against the modern genomes, they revealed no unique derived positions from the medieval era to the current era. And then comparison again against the modern genomes revealed no unique derived positions, arguing that Yersinia was not undergoing genetic rearrangements. And they conclude their story by arguing death may not be due to bacteria, but rather than to the circumstances, either the environment, the dynamics associated with the vector. In this particular case, it would have been the flea, or whether or not it's host susceptibility. And so we have to jump back 660 years and look at the fact that the population of England at the time was very different than modern populations, because they were principally an agrarian society, and they were probably living on many fewer calories than modern humans are living on today. And so they're making the argument that they did not see a substantial difference between the ancient one and the modern ones. There were justifications of how they determined it. The supplemental material goes into greater detail on this, and it's interesting how they compared and contrasted things. So overall, they're making the argument that it's really not different. So they're sort of at a loss of trying to explain the extreme virulence that they saw then to what we're seeing now. But what we learned in the last episode of TWiM, discussing the E. coli K12, it may be the context that the DNA is being presented with, whether it's activation of gyrase or activation of one of these novel histone-like elements that may be effacing the DNA of Yersinia that makes it more virulent.
That's a good point.
In other words, there could be very few changes, which would escape sequencing arguments, and which could make a huge difference, right? That's a possibility. I'd like Vincent to pipe in and tell us a little bit about the history of virus infections which can lead to immunodeficiency, such as measles in nutritionally deprived kids. Mm-hmm. Is it possible that in the 14th century in England there were viruses like it that may have decreased the immune capability of people?
Sure, absolutely. For measles, for example, we believe that it arose about 15,000 years ago when humans first began domesticating animals to eat them. So we think measles came from cows, and as soon as humans were close to them, it transmitted. So it could be measles, it could be something else. Absolutely, that's another possibility, that there was a cofactor. But there are, I think, 97 differences in the sequence from what they got with contemporary sequence, and as we heard last week, only 2 amino acid changes are enough to totally change the transcriptional profile of E. coli K12. So that's a great point, Michael, that it could be one of these changes. So they mentioned that there are some changes in biofilm genes, iron acquisition genes, FOP, for example. So could those make a difference?
Well, certainly iron is a big player in plague. Iron is really a major player in this, and that certainly could have done it, because iron, of course, is one of those materials we derive from our diet, and we need it. And based on the agrarian aspect of medieval Englishmen, they may not have been getting sufficient iron in their diet at that particular point in time, and that may have been one of the reasons it was hypervirulent.
It may be one of the reasons they survived at all, because being iron deficient actually works your way with regard to impeding some bacterial infections from happening. People with too much iron get infected more easily. So in a perverse way, this may have helped people.
Joe, what do you think of all this?
Well, I think it's an amazing study, just technically, that they were able to get the DNA out and study it so completely and get so much genomic information. So I'm still stuck on that part, just marveling over the technology.
One of the things they say in the paper is that because they use this capture method, they could miss big chunks of DNA that may have been present then but aren't present today.
Yeah, for instance, a different plasmid.
Yes. But could you miss a genomic piece also? I guess you could, right?
Yeah, absolutely. I mean, they're only, in a sense, going to get genes that are linked to genes they know about. And so if there were ever big chunks that have nothing linked to today's DNA, they'd never see them. And that's definitely the flaw with the method, that you won't see genes that aren't connected to today's genes. And like you pointed out before, you won't see organisms that you're not looking for. But I just think what they found is just remarkable, that there's so much similarity.
Yeah, it is amazing. But, I mean, I guess you have to use a kind of capture to get DNA from these old samples, right? You can't just do total sequencing.
Not yet. Someday.
It's coming.
Okay, so eventually you'll be able to sequence directly without capture, and then you can see everything that was there. Michael, any other thoughts on this paper before we move on to the next one?
No, I think what Joe said is, I think the beauty of it is they were able to get the old and compare it to the present, and the fact that they saw were able to recover a 4.6 megabase genome, and that's really the utility of Illumina-based sequencing, is it really uses small words of, they were using 55 MERS is what their average sequence read was. And I think that's the key to this methodology, coupled with computational methods in order to assemble a word size of about 50 into a sequence of 4.6 megabases. I think that's what's truly was remarkable is if you can isolate a sufficient mass of DNA and add it to a comparator strain, you can compare and contrast and then really ask what's missing. It's just unfortunate that there wasn't this magic virulence gene that had somehow disappeared from the plague, and then it would, of course, be the stuff of science fiction. The great strain that would take out the world.
Michael, do you want to go at all into this contentious point of this being a nodal organism in evolution? That is, they claim this is the bug that started the great European plagues, and Mark Achtman argues that they're wrong, that this is not the base of the tree, but it goes further back. Maybe we should skip on that. I don't know.
Do you know the answer, Elio?
No, it's a point of contention. I mean, this paper claims that the bug that they looked at is sort of at the base of a tree of strains that then evolved and went on to cause other diseases, other epidemics in Europe. And Mark Achtman, who's an expert on the subject, claims that that's not so. In other words, that this organism is not the base of the tree, but that it goes further back, and the argument has to do with what you include in that consideration and what you don't. And it's really a subject for experts, which it's a little bit beyond me. But I would like to make a different point, namely that Paleo Microbiology, as this may be called, has already had its uses and will continue to have enormous uses. Specifically, as you know, there's been a huge debate as to whether syphilis got started in America and Columbus brought it back to Europe, or it changed in Europe around the time of Columbus, and he is the guy who brought it to the Americas. You know this argument.
Yep.
Yeah.
Well, it looked like maybe there's a solution to it by looking at the teeth of Peruvian mummies and see if they had any syphilis, bacterium, Treponema pallidum DNA in them. Well, the argument is not nearly as strong as here, because obviously they didn't sequence the whole genome or even a significant part of it. But as it stands, as I understand it, yes, they did find T. pallidum DNA in the teeth of Aboriginal Americans, and the idea is that syphilis may have come with the travel of humans over the ice bridge from Siberia to Alaska, and then it changed around here in America, and then Columbus took it back to Europe. Unfortunately, I don't know that this is a definitive argument, and it may have to wait until techniques like have been used here to sequence significantly more than just whatever it was that they were looking at, 16S RNA perhaps. Mm-hmm. But I think this is a subject that is magnificent in its scope, and we can look forward to more and more information, and to be proud in the fact that the answer to enormous historical events may lie, in fact, in contemplating the microbes.
So apparently the first plague was the plague of Justinian, right, Elio?
The first known one.
First known plague.
Yeah.
And so these authors think that that strain is totally different from the one that they're sequencing, right? But that's the point of contention. That's where there's disagreement.
That's the point of contention,
yeah.
Right.
Well, maybe when there are more data, we can get one of these groups to come on TWiM and tell us more about it.
That'd be great.
It'd be fun.
It would be interesting to hear more about what we know about plague from art. Syphilis is a really good example of where there's some interesting hints in old art in Europe. If you look at the old Bosch paintings, the noses of the people in his paintings very often have the classic syphilitic decay. And that would suggest that there was syphilis in Europe before Columbus brought it back, or something very similar to it.
Right.
And I think there's also a lot of art that's very informative about the plague.
Yeah, as this technology gets better and better, we'll get answers to all of these interesting questions.
One other point I wanted to bring up is the whole notion of, we talked about how plague was, devastating to the population of Europe. And the thing that we have not introduced is the whole notion of selecting out a population effect, because if it indeed killed so many people in prime childbearing years, and this goes back to some of the HIV early literature in which they were arguing that people that had the CC5 mutations were less susceptible to HIV infections, and that has since been debunked. And then it was also thought about smallpox actually bringing out these mutations in the human population. And so I thought introducing the discussion of CC5 may be appropriate as we transition to the next set of papers from Science about the host pathogen or microbiome relationship that is going on, and how if indeed the plague bacillus did indeed select for humans with a particular phenotype, if you will, by virtue of not killing them, allowing them to have children. Maybe that is something that was a transacting factor that the humans were making that enabled them to survive, but the plague bacillus still retained that genetic information. And as man has evolved over the last 600 years, we may have effectively changed our spots, so the plague bacillus is no longer as good at being able to infect us.
Right. Our next paper is published in Science this month as well. It's called Linking Long-Term Dietary Patterns with Gut Microbial Enterotypes. What do you think of this paper, Joe?
Well, I really enjoyed this paper. I thought it was a very predictable outcome, but it was nice to have the data to support a prediction that people have been making for a long time. What they found is different enterotypes, and an enterotype, as they describe it, is a profile of the gut bacteria. And so they argue that people will fall into different enterotype groups depending upon a variety of features. But the one that they looked at in this paper was diet. And so they started with a group of 98 people and had them fill out two different surveys which attacked the question of diet two different ways, which is harder than it sounds because people are not very good reporters of their own diets, and that's why they use two different methods to try to triangulate and get some reinforcement one from the other. Then they, and these had enormous detail about the type of food and the calorie intake and the fat and carbohydrate types of the foods that they ate. And then they took fecal samples from these people and looked at the portrait of the microbes in the gut community. And what they found was that they sort of put together in a computer program all the various features of the diet with the features that they found of these communities. And they found that what popped out was high protein seemed to correlate with Bacteroides organisms, one particular genus. And that seemed to be associated particularly with meat consumption because it was also correlated with saturated fat. And those that consumed high carbohydrate diets, the more agrarian-based diets, had high levels of Prevotella, another group of genus of bacteria. And the vegetarians and the one vegan among their 98 were particularly high in Prevotella. And so they defined these two types of communities as the Bacteroides-rich one and the Prevotella-rich one. And they wanted to then ask, could they induce a change that would correlate with what they found in this 98-person sample by changing the diet of their subjects. So they subjected a set of subjects to two different diets, one high carb, one low carb and high protein and high fat. And they actually kept them in seclusion in a hospital so that they couldn't eat anything that wasn't on the prescribed diet. I thought that was a little extreme. And what they found was surprisingly that they did not generate communities that were particularly stable. They didn't stay consistent. And they didn't really resemble the two enterotypes that they found among the 98 people. But what they did find was that within 24 hours of changing these people's diet from whatever they were eating before to the controlled diet, they began to see a dramatic change in the profile of each person's community. So even though these communities changed in different ways, they all responded to a new food source in 24 hours. And that, to me, is the kind of stunning feature of it. That maybe is not surprising to a bacteriologist because we all know how fast bacteria grow. But in the human context, a day isn't very long. And I think we don't imagine such dramatic changes in our organs occurring in a 24-hour period. And this is quite dramatic.
So what does it mean? Maybe you could define for us. What does it mean to have one or another enterotype in your gut?
Well, that's the problem with the paper is we actually didn't learn an awful lot of microbiology from this paper. We don't know mechanisms. We don't understand why there's the change that occurs based on fat or protein or carbohydrate consumption. That's the next set of questions that are just burning to be asked in this field. What does it mean to have high Bacteroides in your gut versus high Prevotella?
So these are gut flora, normal flora, Bacteroides, Prevotella. Do they ever cause problems?
Some of them do in isolation. I mean, there are particular species among those two genera that can be problematic, but more often, from the last few years of analysis, it looks like they're associated with a community type that can cause a problem. So, for example, the obesity studies showed that low Bacteroides and high firmicutes, another group of bacteria, was associated with the obese condition, and they saw that change from a number of different manipulations. So that's not saying that Bacteroides protects you from obesity any more than I think this paper is saying anything about what Bacteroides does for you in the high fat and protein diet. We just know that it's an association at this point.
Right. So I guess the key will be to know, to design a study where you say, does having a Bacteroides enterotype predispose you to certain kinds of diseases? And is it better to have a Prevotella enterotype?
Yes, exactly.
And I can imagine that those are very difficult questions to answer in these kinds of studies, right?
Yeah, they are, because you really can't do you can't do an experiment on the humans. You can't remove all of their bacteria of one type or load them up with bacteria of another type, because that could be unethical if if they're unsafe. And there are enough differences between mice and humans that even the mouse studies for a lot of the metabolic diseases are not completely transferable to people, because the GI tract is different.
Although there are fecal transplants, as they're called, being done with people with Clostridium difficile disease. People there, the gut flora is highly altered originally by an antibiotic, leading to the overgrowth of this nasty bug, Clostridium difficile. And one way to handle it is to take the feces from a neighbor, a friend, a husband, a brother or sister, and transplant those into the person who's sick. So the experiment it's done therapeutically, not experimentally, let's say, and maybe something can be learned from that. I don't know.
And I think those are wonderful therapies, as you say. They're not really experiments, but they are kind of the therapy of last resort for extremely sick people who probably would die eventually, or not in the not too distant future if they didn't have the transplant. The experiments that we can do in humans are kind of interesting, though, because we can treat with probiotics. These are bacteria that have been demonstrated to be fairly safe, and we don't know of any negative consequences of consuming them. And I think it'll be interesting to see if any of the probiotics, or, as this study showed, perhaps even diet changes can be used to induce or protect against particular ailments. And at this point, we know so little that I don't think anyone would have an ethical problem with inducing one community or another in humans, because we don't know enough to say that it's a bad thing to do to people.
But the key is to do a long-term study. You switch someone's diet, say, or give them a probiotic, and then look in the long term, because we know from this study that over 10 days you don't change the enterotype by just changing the diet, right?
Right, and it certainly wasn't a very stable community within those 10 days. So it really was the long-term consumption of one diet or the other that generated the enterotype.
I'm sure you could find people who would change their diet for a year, right?
Yeah. That would be ethical.
Yeah, that's totally ethical, and I'm sure that that'll be done. One of the interesting aspects of this paper were the effects of red wine and aspartame.
Yeah, you want to talk about that?
No, go ahead. You talk about it.
No, you.
Okay. Well, it's one paragraph. They say several factors were correlated with microbiome composition, and that's the important point here. There's a correlation only, and that is consumption of red wine or aspartame were associated with certain compositions of the microbiome. I mean, they didn't, and they found this by looking at these compounds in these individuals' intestinal tracts, not by feeding them red wine and aspartame. That wasn't part of the study. But people who reported, for example, that they consumed artificial sweeteners had certain microbiomes that were different from people who did not. And the same with red wine and people who did not drink red wine. So it doesn't show that this is the cause. There's a good association, but it's suggestive. And you can imagine a study in the future where they would have two groups of people. One takes aspartame and the other doesn't, and you see the effect on the microbiome. This is interesting because alterations in the microbiome, as we've said many times, could be problematic. So you can imagine that, well, in the best world, drinking red wine would be good, and having artificial sweetener would not be, for example.
Yes.
I was talking to one of my colleagues at work about this earlier this week, Ed Baelish, who has done some of the early work using notobiotic animals. And he was describing a study he did many years ago for a private sector firm in which he was isolating the flora from male gigolos and trying to understand if there was some special microbe that they had that enabled them to attract members of the opposite sex and cause this effect. And what
You're
they
serious,
learned
are you?
is that there was indeed unique microbes associated with these male gigolos that would enable them to carry out their profession.
Wow.
Wow, that's incredible.
So the red wine and the aspartame, he didn't find all that remarkable because he said, when you begin to dissect this using the germ-free animal in which you can deposit a defined flora, you can see some of the really neat biology. And I think that's what we're going to have to do in order to design experiments to make some sense out of it. The issue I had with these papers is, while the data were just so beautiful to look at, it was really quite challenging to digest. I think we need a better visual way of discerning the complexities. I was really enamored with the 3 enterotypes, but I really struggled with as Joe was pointing out, the remarkable microbiology that was absent, and I thought the red wine and the aspartame were some of the key morsels in this paper that helped to really put the biology into context.
May I also ask you, the two of you, a question? I'm always a little bit concerned. There's a little caveat in the back of my head about using feces as the samples. I mean, feces are the end product of the story. It's what happens after the food and the bacteria have had an interaction for quite a while, a day or two, over all the reaches, the upper reaches of the large intestine. Well, I imagine that what happens there at the cecum or around there is very different than what you would see if you do feces. Obviously, it's not easy to get to that. You can't aspirate people and get material that you want. But it's a caveat, at least, that looking at feces may not be getting you the whole picture.
Mm-hmm.
Yeah, I agree. And David Relman did some nice work showing that the organisms that are attached to the mucosal lining of the intestine are actually different in the composition. Right. There are different microbes there than in the lumen. And so I think that's probably a flaw with all of these studies, but it's just so much more accessible, and you can get human volunteers so much more easily.
Hmm.
You can always donate at the lab.
Yeah, exactly.
That's a good point.
One of the interesting aspects of the red wine and aspartame was that those have been associated with certain allergies. And I'm always a little suspicious when people say they're allergic to red wine or something like that, because what I imagine is they have some sort of intestinal discomfort associated with eating whatever the thing is. And it's actually their microbes just flourishing in some way that makes their intestines uncomfortable. And it's not really an allergy. But the lore has always been that there's some connection between red wine and aspartame allergies. And it just made me wonder if maybe that's the link, that they affect the same microbes.
Well, I have very good intestines and I like red wine. So that's clearly an endorsement. With N equals 1. I understand that. This is very interesting. That's a really interesting study. But as you say, Michael, the data are daunting. It's really hard to get an overall view of them because they're so complicated. It's not just the microbiome, but they also looked at compounds that are in these individuals They analyze the feces for carbohydrates, fats, amino acids, etc. So this is actually going to be a problem going forward. We're generating all these data by these huge studies, and looking at them is hard. It's not just a gel or a graph anymore. It's a difficult image to look at. Nevertheless, interesting story. Anything else on this before we move on?
No, I think that was it.
Okay. Thank you, Joe. We have a couple of emails before we wrap up. We have 2 emails from our friend Jim in Virginia. The first one has to do with the Yersinia story. The NPR Science Friday topic recently concerned a discussion and debate among several researchers about the Black Plague, and he gives a link to it. The discussion seemed pretty complete, but as a layperson, I'm still not sure I understand how the bacterium are identified using old skeletal material, and question that prairie dogs are the major source of Yersinia pestis in the southwestern U.S. rather than mice. And I also would like to know if flea excrement is really the material that transfers the bacteria to people. Great shows always.
Well, the last point is easy. It's not flea feces. It's the regurgitate which transmits the bacteria. So, wait a minute. But on the other hand, when it comes to the pneumonic, that's quite right. That it's thought that the extent cases of the rare cases of pneumonic plague that we have in the southwestern United States are due to inhalation of fecal material. I'm sorry. I think that may be right.
Michael?
The boo-boos form is you effectively have to get bit by the flea, and the flea's gut is literally chock-a-block full of the Yersinia. And so when you get bit, the flea actually regurgitates the contents of its stomach into you as you are bitten by the flea. And so that's effectively how it comes in. As to the extraction using old skeletal material, as Elio pointed out in the beginning of our discussion about plague, it's hydroxyapatite or the material of teeth or the material of skeleton. Effectively, the DNA is attached to it, and then the method that these individuals used, and if you go and look at their supplemental material, they'll tell you it was principally a proteinase K digestion of the material that then allowed them to liberate the DNA from the matrix that the DNA was attached to. And again, because the word size that they were getting very short pieces of DNA. They weren't isolating 4.6 megabases of DNA. They were isolating little tiny fragments of DNA that were then sticking to these oligos that were impregnated on the array. So it was, if you will, a vacuuming of DNA. And then through the magic of oversampling, they were able to recover the 4.6 megabases. And in reality, that's a function of PCR, being able to dislodge the DNA from that DNA on the oligo that's hybridizing it. So that particular paper is really a primer about how to go hunting for ancient DNA and going after these things.
So what about the source of why pestis in the Southwest? Is it mice? Is it prairie dogs?
Oh, it's prairie dogs.
It's prairie dogs?
It's prairie dogs. It's prairie dogs. And, the old adage is it came to, they trace plague to coming to the North American continent, to the great emigration from China to build the transcontinental railroad, and they anticipate that it landed in San Francisco, very similar to the way it landed in England by ship, and then the rats from the ship, jump ship, literally came to the New World, and then the rats, the fleas were just looking for a blood meal. And since prairie dogs are indigenous in the southwestern United States rather than wharf rats, it just effectively migrated out. Rats don't like the southwestern United States, or wharf rats don't like the southwestern United States as well as the prairie dogs do. And to the best of my knowledge is Yersinia pestis has yet to leap across the Mississippi River. After all, it is the great divider of North America, and it has not yet made it across the Mississippi River.
Okay, Jim has another email. He says, Dr. Schmidt, I think, said in TWiM 12, around the 24-minute mark, that bacterial plaques keep growing, but viral plaques do not. It seems to me, as a person that would both grow to the limits of the dish or until the nutrient is exhausted, another plaque is encountered or the environment changed, dried out, chilled, overheated, etc. Could you please discuss a little more? So what he was referring to, Michael, what you were talking about was bacterial plaques formed on eukaryotic cells by Burkholderia. And you said they continue to grow in contrast to viral plaques, which typically stop.
What the bacteria were doing, and I think we were using it as a metaphor, is the bacteria were acting like Dello Vibrio does in eating microbial lawns. And what was happening here on the eukaryotic lawn is the microbes were moving from cell to cell, and they were effectively expanding out. And as long as the monolayer was present, the microbes continued to expand the plaque. While in a traditional plaque assay, Vincent, as you well know, and you can probably explain it better than I, is you get a viral burst, and then the viruses have to land on a susceptible host in order to cause the viral infection. And by the time things take place, you effectively run out of susceptible hosts, and that limits the size of your plaque.
It depends on the virus and the cell and the combination, but some plaques will continue to grow as long as there are nutrients left in the medium. Others stop at a certain point. So it really depends. So some viral plaques will keep growing and some will stop. I don't think there's any generalization that you could make about all viruses in that respect. The next one's from Michael, who writes, Hello, TWiM and TWiV team. I wanted to say that I very much enjoyed podcast number 6. I'm a great fan of phages myself because they introduced me to biology in general. I used to have a very poor understanding of biology. Our school thought skipping biology and using the time for math instead would be better. But since I first glanced at a bacteriophage virion of T4 a couple of months back, it created a spark in me and I wanted to know everything about him. Well, I have and still have to study biology from scratch, but I think I made in this short time great advances. Your podcasts, TWiV and TWiM, are excellent and provide entertainment and education. Also, thanks to the fact that I live in China, and here it is very cheap to buy reproductions of scientific books, $5 in China compared to $100 or more in developed parts of the world, I could provide myself with very good learning materials. Would love to hear more about phages in the future. Also, I would recommend a book. It's called The Bacteriophages, edited by Richard Callender. It describes many aspects of phages and is absolutely perfect for anybody who wants to learn more about them in detail. Also describes many aspects of phage therapy. It's not written for the bio-noob like myself, but thanks to Google, I could learn everything I didn't understand along the way. Greetings from China. P.S. I always listen to your podcasts on my phone in the kitchen while I cook. So every time there is a rather long TWiV or TWiM, my wife knows what I'm going to cook will be good, since I will spend at least 1 hour in the kitchen. So I guess thanks from my wife, too.
Can we get takeout?
Yes, really. Sounds good. All right, the last one is from David, who is a microbiology student at NC State University, North Carolina State. And he says, is it possible to create a liquid culture that simulates the natural conditions in which Pseudomonas aeruginosa can survive? I was hoping that one that simulates mammalian lung tissue might work. I don't know the answer. Do any of you know?
Well, I think it's a little bit unlikely in liquid culture because Pseudomonas forms biofilms on the surface of the lung, and I think being submerged or even floating in liquid culture would be a very different habitat because it wouldn't be attached. But I think that, there are some in vitro assays with lung tissue, and I think somebody was using even artificial lung tissue. As long as they have a surface, I think it's probably more similar to the natural infection.
It depends a little bit what question you want to ask. It really matters if you're asking a question that deals with the pathogenesis, in which case I think what Joe says is absolutely right. It doesn't make sense to look at something in liquid when it's not happening there.
I have one more email. It's from Don. He writes, could you do an episode on Wolbachia? It seems to control the reproduction of Drosophila, mosquito, etc. Does its influence extend to other, I don't know what he's writing here. He writes
multicellular, maybe?
I don't know. But Don, I'm going to refer you over to...
You think he means multicellular?
Insects, he means.
Could be. He's written multicultural and then verts as well, so I don't know what he's got here. It's mainly in insects. A good fraction of the world's insects, arthropods and insects, are infected. And we actually talked about it on TWiV. I'll put a link to a TWiV episode where we talked about an interesting study where they're using it to try and control dengue virus infections. But I'm sure at some point in the future on TWiM we'll do another one on Wolbachia. 2 other brief comments. Was there any follow-up to using metallic copper in hospitals to control the spread of drug-resistant microbes? And there you go, Michael.
The answer to that question is yes. We just released the results from a clinical trial that we conducted at 3 hospitals in the United States: Memorial Sloan Kettering, the Medical University of South Carolina, and the Ralph H. Johnson Veterans Medical Center, where we were able to show that copper surfaces significantly lowered the rate of hospital-acquired infections in medical intensive care units. And the simple take-home message is there was about a 41% relative risk reduction in the number of infections that developed in patients that were treated in a room with a limited number of objects that were surfaced in copper versus the control rooms. And so those studies will be published shortly. So you can, once the paper is out, we'll have more to say about that.
Thank you, Michael. And then finally, he refers to Draco. I don't know if any of you have heard of Draco, but it's the name of a protein that has been made at MIT, which has a rather broad antiviral spectrum. And so he writes, although the Draco protein was undoubtedly made in a Slytherin lab, does not Hogwarts code require a balanced wait-and-see approach? I bet you are a Ravenclaw.
Ravenclaw. I'll have to ask my colleague's daughter, who is the world's foremost expert on Harry Potter. She'll effectively divine whether I'm indeed a Ravenclaw.
I mean, the Slytherins are nasty, right? But I don't know what the Ravenclaws are.
Yeah.
So the Draco, we had panned the Draco protein in TWiV, and he's saying wait and see. And you're absolutely right, Don. Maybe that's the way to do it. So would a Ravenclaw wait and see? I don't know. Anyway, thanks for all the letters. We do this every other week, and you can find TWiM at microbeworld.org/TWiM. You can also stream the episodes to your iPhone or Android device. There is an app for that over at microbeworld.org/app, made by the American Society for Microbiology. And we always love to get your questions and comments. Please send them to TWiM at twiv.tv. If you do listen to TWiM on iTunes, please leave a review. It really helps us to stay on the front page of the Medicine Podcast directory. Joe Handelsman is at Yale University. Thanks for joining us, Joe.
Thanks for having me. It was great, Vincent.
Always a pleasure. Michael Schmidt is at the Medical University of South Carolina, and Elio Schaechter is at Small Things Considered. Thank you, Elio.
Oh, my pleasure, as always.
I'm Vincent Racaniello, and you can find me at virology.ws. Thanks also to the American Society for Microbiology for supporting TWiM, Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for technical help. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.